So this evening, I'd like to welcome Dr. Frank Ashwood to talk to us about soil biodiversity monitoring in the UK. He's going to give us a little tour of soil biodiversity and then go into the technicalities of monitoring them. So I'm personally super excited about this. Can't wait to hear more, Frank. So over to you.
Great, thanks, Kieran. Yep, so I will dive straight into it. Hang on, there we go. Okay, so yeah, thanks for that introduction, Kieran. I won't say too much about myself, just to say that yeah, I'm a soil ecologist. I was in the UK, but as Kieran mentioned, I'm now based in New Zealand. So kia ora and good evening to the UK and good morning from over here. My background is looking at soil invertebrates and their ecology in woodlands. And for my sins, I've been a member of the Earthworm Society of Britain for a while now. So if anybody there is infused about earthworms by the end of this talk and other soil invertebrates, you can, if you're not already a member, you should go look at that website and sign up, and you get lots of information there. I'm also a macro photographer of soil animals, as if researching them wasn't enough, and I'm going to use quite a lot of my photos in this presentation. The way that this talk will kind of run is I think I've got about 40 minutes. I'll try not to overrun too much. I'm going to assume that not everybody here is a professional soil ecologist. So what I'll do is I'll spend the first chunk of it taking you on a bit of a safari through the soil. So we'll go and meet some of the organisms that we might like to monitor, and I'll talk about a bit about what they do. And then in the back half, we'll go into methods for monitoring soil invertebrates. So if anyone gets infused by this and wants to give it a try, you can. And then I'll wrap up by talking about some of the major current monitoring initiatives that are out there. So without further ado, a good place to start is probably to ask ourselves, what is soil biodiversity? And you could ask 100 different people and get 100 different answers. But the way that I like to look at it, and seems to be commonly accepted, is just it's all of the animals that, or the organisms which live in soil. So if an organism spends part of its life in the soil, I like to think it's a part of soil biodiversity. And there was a recent study by Mark Anthony and colleagues. I didn't put the citation here, sorry, but yeah, you should look that up. That estimated that actually soils contain roughly half of the biodiversity of the Earth. So they're a major global biodiversity reserve, and thus it's really important that we monitor this. Why we're interested in them? Well, of course, all biodiversity has inherent value, and that's a reason enough. But also, from a purely selfish perspective, a lot of the functions and the things that we get from soil, or we rely on soil from, so carbon sequestration, nutrient cycling, plant growth, floodwater infiltration, all these services that we rely on, our food, are largely impacted or regulated by the interactions between soil organisms. So it's really important that we understand this fully. And of course, scientists like to categorize things up into nice, neat little parcels so we can study it. And there's lots of ways that we could do this with soil organisms, and I'll talk about a few of those today. But one way that's commonly used, for better or worse, is to do it based on the size of the organisms. So we have large creatures, which we call megafauna. Then we have macrofauna, which are smaller than that, so about 2 millimeters in body width or larger. And then we have things a bit smaller than that, so we call those mesofauna. And then right at the very smallest, less than 0.1 millimeter in size, we have the microfauna and microflora, so things like fungi and bacteria. And I'll use that kind of general size scale as a framework for the safari we'll go on. And I've said that there's a lot of biodiversity in soil, but how much life are we actually talking? This is kind of a generalized schematic, but if in theory, if we were to take about 1 meter squared of a healthy soil from a woodland or a pastureland or something like that, and we excavated all that soil and we were somehow able to extract all of the organisms from there, this is something that we might find. So you might find maybe 300 earthworms or so, but you might find thousands of their small relatives, the potworms, Enchytrades. As you move down in scale to smaller stuff, though, you get even more of it. So we might find perhaps 50,000 springtails in there. We might find 100,000 mites. Smaller still, 5 million nematodes. And then if you go right down to the microbial scale, I think that's 10 trillion. probably. It's a kind of a nonsense number, really. But yeah, it's a lot of life in the soil, and thus worth plenty for us to monitor, I guess. So starting at the big end, we'll go on our safari now, and I've only got one slide devoted to vertebrates because this is going to be largely an invertebrate-based talk. But just to say that in the UK, there are some megafauna in the soil. They're mostly predatory. So I guess there's an argument to be made for rabbits being a herbivorous soil fauna, but with their burrows. But generally, most soil vertebrates or soil megafauna are predators. They regulate the populations of all of the other invertebrates in the soil. And two of the major ones that we have in the UK would be something like the slow worms and also the mole. So moles are major earthworm predators, and they can have huge impacts on earthworm populations in the field. If you've ever seen a field full of molehills, you can probably appreciate that. They do actually, it's worth noting, they do have a special burrow in a special chamber in their burrow, which is basically like a larder for earthworms, which they paralyze by biting them and severing their nerves, and then stuff them all into this larder so they keep fresh to eat later on. So moles are probably the ones that we need to consider most in this category in the UK. But we're going to move on because all the exciting stuff, I'm biased, but I think the exciting stuff's in the next couple of categories. So if we go down now to the macrofauna, so these are organisms which are greater than 2 millimeters in width, but smaller than the megafauna. There are also predators at this level, and these are things we might like to monitor or be interested in. So sorry, I've got a terrible cold at the moment if I keep coughing. We've got some major predators in the macrofauna. These are three of them. So ground beetles, predatory beetles, are quite important. Ants are kind of omnivorous, but they do have a big predatory role. And of course, centipedes are a major group of interest. And personally, they freak me out a little bit. Of all of the invertebrates, they're the ones I like the least, I would say. But this one was quite an impressive specimen, Critops anomalans. It's one of the bigger species that we have in the UK. And they're distinguished from millipedes. If anyone's ever got confused between the two, they've only got one pair of legs per segment. And they've also got a predatory function, whereas millipedes are a bit more sort of detritivorous. And they're noteworthy for this really robust, intimidating pair of sort of fangs that's underneath their head. They're actually not fangs, so if you ever get a chance to look at them, they're actually the first pair of legs which have evolved over time to form a kind of fang-like purpose, but they inject venom into their prey, which they use to immobilize them for eating alive. Moving through the macrofauna now onto the detritivores. We've got two of the major groups that we could consider in the UK are the woodlice and the millipedes. So woodlice are pretty important decomposers or detritivores in high organic matter situations, and you'll see that if you ever flip over a log or anything, and just absolutely full of woodlice. These ones are particularly beautiful ones. This is Platyarthurus hoffmannseggi, the ant woodlouse, and they spend their entire lives within ants' nests. So if you ever look inside a yellow meadow ant's nest or something, you'll probably find these little white woodlice running around. They eat all of the organic waste that's produced by the ants, so they keep the ants' nests clean. There's some pretty beautiful woodlice in the UK, actually. So we've got rosy woodlouse here. These are associated quite heavily with sort of limestone situations. So you find them in caves and under limestone rocks, but you also find them in churchyards where they eat the mortar on old churches. And I'm still yet to be convinced that there's anything cuter than a baby millipede that we've got up the top right here. They're frankly adorable, but they're also incredibly important detritivores. So all of these groups of organisms will be processing organic material, decaying wood and other decaying organic material, and chewing it up and passing it out so that it's primed for microbial decay and nutrient release. And they process huge amounts of carbon in the ecosystem. And of course, I had to mention earthworms when we're talking about macrofauna. I could give a whole talk on them, but I won't. I will control myself. And Kieran's got some exciting ones lined up next year that you should go to. But just to say that earthworms are, of course, really important detritivores, or I put omnivores here because there's evidence that they might actually eat bacteria and fungi and protists and other things when they're ingesting soil as well. So, but they process huge amounts of organic material. They're really important for soil structure, for regulating air and water infiltration into soil. We call them ecosystem engineers. Darwin called them nature's plow for a reason. So yeah, come to some of those talks and learn more about them. I don't have time to go into them in huge detail today, but just to say that there's quite a great diversity of earthworms in the UK. There's about 31 species, and they do all perform semi-different ecological functions. Now, I'm not going to take this with a pinch of salt, this diagram, and I'll talk a bit about the ecological category concept later on when we're talking about interpreting monitoring data, because it's not necessarily quite so simple as sort of the deep burrowing, shallow burrowing, surface living groups that we tend to typically talk about. Although it's kind of a useful way of thinking about it, it can be a bit more complicated than that. And we know that, for example, there are earthworms that exist outside of the soil completely. So here we have Dendrobina attempsi. This species is associated with trees and decaying wood. So this was found in a piece of deadwood, but also obviously we've got composting earthworms like Dendrobina veneta here as well. So and the way that you might like to sample those might be different depending on what your monitoring method is that you're undertaking. So we'll come to this in a bit. So that's a quick run through of some of the macrofauna, and now we'll go into the mesofauna. So these are organisms that are less than 2 millimeters in size. And this is stuff that you can still see with a hand lens and the naked eye, but it starts to get a bit more challenging. And then past this group, you really need a microscope. So coming off the back of the earthworms, we should talk about potworms or enkydrateds. So if you ever dig up a high organic matter soil, or you look in some deadwood or just some decaying organic materials, you might find a lot of small white worm-like creatures. And if they may be nematodes, but they're probably potworms if you can see them easily. And these are annelid worms. They're related to earthworms. They basically perform similar functions to earthworms. They're just a lot smaller. But you do tend to find them en masse in situations where earthworms are a bit less abundant. So possibly in like more acidic soils and very high organic matter or humic soils. And they can occur in great numbers. So, in a square meter, you can have maybe, you can have a few thousands of these. And if you don't have earthworms there, then these are probably performing very much the same function, but just very much on a smaller scale. Particularly like this photo, you can actually see what they're doing. They're just processing all this. This is their gut. And their bodies are pretty much transparent. So you can actually see the organic material passing through the digestive system of the potworm, and they're just doing this 24/7. So you can imagine that actually, although they're small, they do end up processing a great deal of organic material and soil through their bodies, which is helping to prime it for microbial decomposition. Moving on to the predators, the mesofauna. So these are, and apologies if anyone's phobic of arachnids, but here's a massive pseudoscorpion for you. These pseudoscorpions are probably one of my favorite groups in the soil. They're just very cool. They are pretty aggressive predators for their size. So if you're a springtail or something, this is your worst nightmare. They perform a very similar function to normal scorpions. I mean, they're arachnids, they're related to scorpions. They differ in size and also the fact they don't have a stinging tail. Hence the pseudoscorpion. They do look like them though with these pincers, but they do still deliver a venom to immobilize and partially digest their prey, which they deliver through a special gland that's attached to their pincers. So when they grab something with their pincers, they'll inject, say, a springtail with venom, and it will start to digest the insides of the springtails, which they'll then sort of chew up and drink out like a soup. And speaking of springtails, so this is one of the major groups we might like to consider if we're looking at soil biological monitoring. And there's a few different orders of springtail, so I'll run through those. But they seem to be food for quite a lot of the predators in the soil, as do earthworms. And but they're incredibly important. They are widely abundant. After mites, they're probably the most abundant and diverse group of invertebrates in the soil. Sorry, arthropods in the soil, not invertebrates. And yeah, they perform a lot of functions. They graze on fungi and microbial coverings on decaying wood. Some may actually chew on decomposing organic materials itself and help to break that down. So in that way, they help to regulate microbial populations and nutrient cycling. They're pretty interesting creatures just in their own right. They can be incredibly beautiful. Some could be covered in these sort of metallic iridescent scales. They're often quite hydrophobic. They've got really interesting structures on their cuticles, which mean that soil and water don't stick to them. And materials engineers have been sort of looking at those to try and improve material engineering science for creating waterproof and anti-spoiling surfaces. And as an example, here's one walking on the surface of some water here. They've also got springing organs, which is this kind of forked tail-like appendage that is under the body about here, and it is held under tension against this sort of little gluey peg that holds it under tension. If they're disturbed, they will flick that into the ground and launch themselves up into the air to get away from the danger, hence the name springtail. And there's different types of them. So those with the sort of slender or elongate springtails before. These are the globular springtails. They are incredibly adorable organisms. Everyone always seems to think of these first when they think about soil organisms. There's a New Zealand Entomological Society is actually running a competition for New Zealand Invertebrate of the Year next year. And one of the candidates is the globular springtail. And I encourage everybody to weigh in on that. And in much the same way as John Oliver did, I think we should probably overload it with the globular springtails, because they deserve a lot more credit than they get. In fact, some of these species are the ones that have been shown to launch themselves up to 100 times their body length in height to get away. So I think that's equivalent to you or I just suddenly jumping over the Eiffel Tower, which obviously is quite amazing. And some of them can actually live in lightless scenarios. So this is a cave living species. Yeah, they're food for a lot of predators, and they're also important regulators of microbial populations. And this is the final group that I'll mention. These are the podromorph springtails, podgy springtails. They look like little gummy bears, basically. The majority of them are actually kind of pale. They look like this in the bottom right here. There's a huge diversity of them. They're probably my least favorite group of organisms to identify because it's hard. They can be quite small and there's not a lot to go on sometimes. Quite often they're eyeless, they are pigmentless, they may not have a springing organ. So a lot of the soil living species that live deeper in the soil layers will have just evolutionarily lost their springing organ. There's just no point to it. And that in itself is something that we might be interested in when we come to assess these populations. But some of them can be incredibly beautiful. So the ones that live closer to the surface can have such nice coloring as this, and they can be very ornate. But they are food for, as I said, quite a few things. And there is a lot of mites, predatory mites, that will see those organisms as a pretty tasty snack. So here we have a selection of some of the predatory mites in the soil. Mites being arachnids related to spiders and the pseudoscorpion that we saw earlier. And it's worth noting that mites are probably the most abundant and diverse arthropods in the soil. So it's just an overwhelming diversity of mites, and there's not enough people looking at them. So if you want to get into mites, then you should definitely, I encourage you to do so. And Matthew Shepherd at Natural England is someone that you should be looking up, because he's produced a key that will help you identify to family British mites, which is a really useful resource. So here we have a selection of predatory mites. This is a snout mite, a bedelid. These are all active predators, so they'll be running around the soil surface looking for springtails and things to eat. These bedelids will pierce springtails and other prey with this pointed mouth part, and they drink out the fluids. Very similar to here with this ragidid mite. So this is a really beautiful prostigmatid mite that you'll see quite often running around on logs or on the surface of the soil. And they're always just endlessly chasing down prey. And here we have a whirligig mite. So this is in the family Anistidae. And you'll see these a lot if you ever in the summer out in a garden or a park or something, and you see like a wooden post or a fence, and you see lots of little red mites running around really, really fast. It's probably one of these whirligig mites. This is what they look like up close. They're incredibly fast, and there's actually a species of anistid mite in North America, which has been clocked as the fastest organism on Earth for its body size. So relative to its own size, it can run, I think it was is it 120 or 200, something like that, times its own body length. So it's basically the equivalent of you and I outrunning a commercial jetliner is the speed that it goes to. So really amazing organisms, and a nightmare if you're a tiny invertebrate. Mites have got a huge range of trophic or like feeding mechanisms, which is of interest to us if we're looking to monitor mites. Here we've got a selection of just parasitic forms. So this mite on the left here is actually a nymph stage of a mite that will be a predator when it's an adult. But as a nymph, it's a parasite, and it's feeding on the hemolymph, the blood of a harvestman's leg. After a while, it will drop off and molt and assume its adult stage as an active predator, much like the previous mites. These mites on the right here are Ricarduella udemansi. This is the white slug mite, and yeah, they spend their entire life living on or in the bodies of slugs. So I don't think it's nice for them, and it's certainly not nice for the slug. But pretty much every slug in the UK will be infected with these. So if you ever get the chance to look at a slug up close with a hand lens, you'll probably see little white dots running around. It will be this mite. For a long time, they were thought not to be harmless, but actually it was then realized that they are feeding on the blood of the slugs. So they will be weakening the slugs over time, and they actually reproduce inside the mantle, inside the lung of the slug. So here's the photo I took of one emerging from the breathing pore that when it opens and closes on the side of the slug. So it's a pretty grim existence, to be honest. There are herbivorous mites. So I said mites come in lots of forms, and they have lots of feeding strategies. There's not a huge amount of them are plant pests, but there are some. And this one, Penphleus major, the blue oak mite, is a agricultural pest. Noteworthy for having a dorsal anus. This pore here is actually its anus, so its bottom's on its top, and they spend most of their time just feeding on the fluids of plants, so they could be hanging upside down. So you can imagine that actually it's quite useful if you're hanging upside down on plants all the time to be able to just drop your waste directly downwards and not risk covering yourself, because it's probably got lots of sticky sugars and stuff in it, and you don't want to get all gummed up. From a source perspective, a really interesting group are the fungivorous mites. So these are typically prostigmatid mites, but there are a few other sort of more hard-bodied mesostigmatid mites that will feed on fungi and regulate microbial populations in the soil and decaying wood in that way. These ones are particularly interesting. They're very common, Linopodes species. It looks like it's got some antennae, but of course it's an arachnid. They don't possess antennae. These are actually the first pair of legs which have sort of developed this kind of sensory function. They're very long and they wave them around. I think they also use them for communication, because quite often you will see groups of them sort of collected together, waving their legs at each other in the air. Who knows what they're talking about? But they can be pretty beautiful, and they will be feeding on the fluids of slime molds and other fungi. And then just to sort of wrap up, there's a few other groups of mites which are particularly interesting to us from a soil monitoring perspective. So in terms of bioindications, so one of the major groups are the orobatid mites. And so these are hard-bodied mites which have a K-selected lifestyle, life history, which means that they are slow-living, very low reproductive output. They take a long time to develop into adults, and they invest quite a lot of their energy into armor and camouflage. And they kind of develop these really elaborate anti-predatory devices, so like all these wings for tucking their legs under, or really hard sclerotized body cases. Some even invest in some effort into making camouflage, so that they can stay alive long enough to actually eventually reproduce. And I'll just give a shout out here to this species, Platinothris peltifer, quite common in the UK and generally around the Northern Hemisphere. It's noteworthy because it's developed this kind of interesting approach to its reproductive output, which is that it's a parthenogenetic or parthenogenetic species. So they reproduce entirely by cloning. There's only female individuals of this species, and they've been doing that for about 100 million years. So since before the dinosaurs, or around the time, before the dinosaurs were extinct, they have been reproducing asexually. So the females just had enough of the males in that species and decided to just do their own thing. Can't blame them. And here, the dust mites are the final group I'll mention, but they are the complete opposite to those orobatid mites. And I'll come back to those two groups shortly when we talk about monitoring methods. But they're very interesting because they've got the complete opposite approach. They are generally selected, which means they've got rapid, like R for rapid, dispersal, rapid life cycles. They don't live very long. They have huge reproductive output, and they have this kind of boom and bust kind of scenario to approach to life. Whenever a new resource opens up, say some soil is disturbed, they can get in there quick and make use of that organic material. Quite a lot of the pests we might find in sort of food industry and on some cheeses and stuff, for example, would be these mites. So they can be a bit of a pest. And if anyone here is a microbial scientist and you work in the lab, I think quite often if your agar gets contaminated, you might find a bunch of these astigmatid mites running around in it. But they are dispersal specialists. They can get around, and they get around very well. They can't get blown in the wind or something with these long hairs, but they also have specialist, some of them have specialist life stages when they're nymphs just to get around. And we call that foracy, when something hitchhikes. And so these mites here are, this one here and this are a special phoretic stage in the nymphal stage of the astigmatid mite. And what they do is they clamp onto other organisms. So here there's one on a millipede's leg, and here there's one on the back of a different mite, on a mesostigmatid predatory mite. And what they do is they lose their mouths, they lose their anuses, they get rid of those, don't need them. They're just going for a ride at the moment, and they develop special sucker pads, and they just affix on like a limpet onto these organisms, and they go for a ride and then drop off later on. Who knows what causes them to do that, and then turn into an adult and go about their life like this in the soil. And so they're very able to move to new places and exploit new resources. And this is important. This and the previous group are really important to consider when looking at soil disturbance. And I'll come to that. Finally, it was in the title, so I should really mention pauropods. I haven't got a huge amount to say because nobody really looks at them. If anyone here wants to spend more time on pauropods, then you really should. There's a chap in the UK called Dawid, who is looking at these and developing the keys to them, and hopefully will encourage more people to be able to identify them. They're related to millipedes and centipedes. They're very, very small versions, essentially, but they're kind of mysterious. Nobody's really looking at them. There's not much money out there to pay people to look at these obscure groups. But they're incredibly beautiful little creatures. They're probably fungivorous and detritivores, and they've noteworthy of having these what we call biramus antennae, these branching, very alien-looking antennae, completely laden with sensory functions, and they can come in a broad variety of shapes and sizes. But these are some common British species. They're pretty small, but once you get your eye in, you can find them with a hand lens, and I encourage people to go and do that. They're very common on sort of, well, not common, but if you go to woodlands and you flip over rocks or logs, you should see some of them, and more people should be looking at those. And if once you've got your head around pauropods, then you should really consider proturans as well. Absolutely nobody is looking at these. They are, we think, fungivorous or detritivores, so they'll be feeding on detritus, decomposing organic material, whatever. Although in lab cultures, they've been seen to feed on fungi, fungal hyphae, but generally no one's observing them in the wild, so we don't really know for sure what they eat and what they do. They can be very small. They're sort of less than 1 millimeter in size generally, maybe up to 2 millimeters if you find a really big one. We call them coneheads for short, as a common name. They've got this kind of conicular head here. They're actually hexapods, so they're related to insects and springtails and bristletails and some other things I haven't been able to have time to mention today. But they hold out, they don't have antennae, they hold out these two front legs to sense their world, and they don't have eyes either. So they're this very cryptic group that we just don't know enough about. And they do pop up in soil samples though. And then very, very finally, we're going to wrap up by talking about the microfauna and microflora. So just honorable shout out to the nematode worms. I'm not a nematologist, so I won't dwell too long, but just to say that they have got a huge variety of feeding mechanisms. You've got parasites, fungivores, herbivores, bacterivores, everything. You name it, there's a nematode for it. They also do foracy for dispersal, so they hitchhike. And here we've got two nematodes curled up taking a ride on the back of a predatory mite. Definitely one to look for if you're doing soil monitoring. And then finally, just to give a shout out to the bacteria and the fungi. I'm not going to go into them in any depth today, but they are incredibly important for cycling nutrients and carbon in soils and for soil structure. And there's something definitely that our DNA interested friends will be looking at a lot more. So there's your safari. Hopefully you're interested in soil biodiversity now, and I'm going to quickly go through some methods and then some monitoring initiatives. Now you want to do it, this is how you do it. So just to quickly say, obviously soil is a very complex environment and there'll be lots of things living at different places. And so the methods that you will adopt when monitoring soil biodiversity will have to be dependent, will be appropriate to what you're looking for. So you might look at different layers in the soil, you might look at associations with vegetation, you might want to look at microhabitats. You might even want to look at suspended soils up in trees. There's lots of soil invertebrates that will be living outside of the soil. And then what you're looking for will determine what kind of sampling method you might adopt. You might want to use something like a pitfall trap if you're looking at surface active invertebrates. You might like to have a subsoil kind of subterranean sampler, which is like a pitfall trap in the soil if you want to catch things that might move through horizontally through the soil profile. You might want to take grab samples of suspended soils. You might want to do various things. So something to consider. If you're looking for earthworms, one common method that we do is to dig a soil pit of a known area. So this is 0.1 meters squared, this quadrat. You excavate all the soil around that from in there and for earthworms. And then what you might like to do is also pour a vermifuge solution. So this would be a mustard water solution into the pit to extract all of the deep burrowing earthworms that you haven't been able to capture with this. You might also like to look in deadwood. As I said, earthworms and other invertebrates live in microhabitats, and that's something I would encourage people to do responsibly. Don't destroy every bit of deadwood in your local forest, but you will find some interesting invertebrates there that you won't find in the soil. And if you want to do a non-invasive kind of sampling method for other invertebrates, microarthropods, you might like to do something like this, put a pad. This is just like a Brillo pad, and you can put that in tree hollows and branch nooks, leave it for a while, and then bring it back in and put that into under a lamp to extract the invertebrates from that. And that's a non-destructive sampling method you might like to adopt. However you've done it, once you've got your sample, if it's earthworms, you've got your earthworms already, that's great. If you're looking for microarthropods, you'll want to put your soil core or your Brillo pad or whatever on one of these. And this is a homemade version I did years ago. You can do it relatively cheaply at home. You just need to get some funnels, put it on a bit of mesh on top of the funnel and put your sample on there. And then underneath have a pot of ethanol waiting to collect them. You can just have a cup without any ethanol underneath, but be aware that if you're collecting predators and their prey, unless you collect that sample pretty quickly, you're probably going to lose a lot of the prey species in your pot. So we tend to put ethanol in there just to kill everything and fix it so that we can look at it under the microscope. And you leave that for about a week and you'll come up with something like this. So this is a sort of a woodland soil microarthropod sample. Lots of orobatid mites and predatory mesostic mites. You've got lots of those little white springtails in there that I mentioned before. So yeah, you've got your sample there. If you're looking for nematodes and egg trays, you do a very similar thing, but you have water in your funnels, and then you will collect the sample at the bottom, put a little clamp on a tube, and the worms will move down through the filter paper and into the bottom of this tube, and you can siphon that off and look at them. So you've got your samples. What are you going to do now? So you've done the first part of the monitoring. Now you need to collect some data. So I'm a big fan of morphological identification. There are some pros to it. There are also some cons to it. So morphological ID is just basically looking at features down a microscope to determine an identification. You can do it to varying taxonomic levels depending on what the intention of your monitoring initiative. You can also collect quite a lot of interesting data that way. So you can get abundance data by counting the organisms. You can get biomass data if you weigh them. It's particularly useful for earthworms, but you can do it with other invertebrates. And you can also measure functional traits of the organism. So you can look at specific parts of the organisms which are related to their life in their environment. And I'll come to that in a minute. So those are all really interesting things. But of course, there are some technical challenges. You need resources. It takes a lot of time, takes a lot of personal skill to get up to that level. And it's also dependent on the availability of keys. And I mentioned before for mites, there's a family key. You should look up Matthew Shepherd, Natural England. Well, here he is, and here's his key. So that's something to consider. The alternative, and a lot of people do this, is to look at DNA. So you can take either your Tolgren funnel extract of your invertebrates and run that, or you can take just soil, fresh soil, and process that in the lab and compare the DNA reads that you get against the barcodes in various taxonomic genetic databases and arrive at species determinations that way. But of course, that's got pros and cons as well. It's expensive, but probably not quite as expensive now as someone's time looking down the microscope, unfortunately. But you can't necessarily get abundance data or biomass data. You can get kind of analogs of that, but there's pros and cons. And of course, your results are only as good as the barcodes that you've got in the library. And so really, ideally, I think these two methods should happen together in tandem. And in fact, that's what's happening in a lot of method monitoring initiatives now. There's also machine learning approaches. So just quickly to mention a couple that we've been involved in the UK for its research, where I was working before. So here, you can either take a scan of a petri dish with a community on a flatbed scanner, or you can actually use a digital microscope, digital camera with a macro photography lens, take high resolution imagery of these organisms and use machine learning and AI to actually train the algorithm to do the identification for you. And these methods are actually getting quite impressive now in their accuracy. And one of the pros of this is actually it can really speed up the time that it takes to process samples, which is the major bottleneck with morphological ID. So you've got your data through those methods. How do we interpret that for monitoring? So I'll talk about a few different methods, and then I'll wrap up by talking about some of the initiatives that are happening in the UK and around the world. So you've got your data. How might we interpret those soil biodiversity data? Well, there's a whole swathe of traditional ecological metrics that we might like to use. So you might look at the presence absence of specific species. So if you know the environmental tolerances of different groups, so earthworms have a certain pH range that they can tolerate or not, for example, you might start looking at those as indicator species. You might also look at species richness, diversity, abundance and biomass. So these are all typical metrics that ecologists will use, and it's exactly the same for soil invertebrates. You might like to consider, though, that comparing different land use types with that, actually you are making some assumptions. So be careful about how those are interpreted, whether you're just assuming the increased abundance and biomass or diversity of earthworms, for example, in a field or between fields, means that there's an improvement in habitat quality between those fields, because that's not always the case. And of course, these things can't increase exponentially. You might reach a point where you've reached the maximum carrying capacity of that environment. So it's always worth being mindful of what you're looking for and why. But you might also be interested in taking things a little bit further with the indicator species approach. So for example, here are two of the mites that I mentioned earlier. You've got the orobatid mites and the astigmatid mites, those dust mites. They've got very different life strategies. So these were the K-selected, very slow living, slow developing, low reproductive output mites. Then you've got this fast boom and bust kind of astigmatid mites. And these can actually be really informative for looking at disturbance situations in soil. So I was doing some research on a reclaimed landfill site, and we were able to identify that the reclamation hadn't been done necessarily to a particularly good standard, because the soil had been incredibly disturbed. There was absolutely none of these orobatid mites present whatsoever. They just can't handle disturbance. Soil disturbance, it kills them, and they haven't got time to rebuild their populations. And I think almost entirely all of the mites that we found on that site were these astigmatid dust mites, which indicated that the soil quality, the soil condition was incredibly poor. So that's a very sort of rough and ready way of doing it. We might also look at approaches like functional traits. So this is something that's emerging now as quite a popular approach to interpreting soil biodiversity data. And it's complicated because it requires a whole new swathe of measurements to be taken, which we're not normally doing. You need to look out for specific parts of these organisms and measure them, measure their length and presence absence, rather than just looking at a species. But does give us an idea of how these organisms relate to their environment and their specific adaptations to the environment they live in. So, for example, in the springtails, the collembola, these two would be more surface living springtails, and these are deep soil living springtails. So here we might be measuring things like presence or absence of eyes for detecting light. We might be looking at the presence, absence of a jumping organ, this furca, this spring that puts the spring in springtails. We might be looking at presence of pigmentation in the skin, in the cuticle, and these are all indications of organisms that live on the springtails that will live on the surface. Whereas here, you will find that these springtails won't have ocelli, eyes, they won't have pigmentation, and they've probably got a very reduced or a completely absent springing organ. And by measuring those, we can actually start to interpret the ecological functions and the areas in which these organisms live in the soil, and potentially the roles that they adopt within the soil. And by utilizing that kind of approach for earthworms, some scientists, Bottinelli et al., were able to sort of revolutionize the way we interpret the ecological categories of earthworms. So on the right here is the triangle that we normally kind of think of when we look at earthworm ecotypes, ecological categories. So here we've got the sort of shallow burrowing earthworms, we've got the surface living earthworms here, and we've got deep burrowing earthworms here in these corners. But then we actually realize that a lot of these earthworms live in intermediate categories. And this is something that I think is going to be done more for British species. This is more of a European study. But it helps to more inform our understanding of the ecological functions of these organisms. So that's something that's being done quite a lot now. We could look at the trophic guilds of organisms as well as a way of categorizing them. And that's something that I was doing in that soil safari we were on earlier. I was presenting their feeding mechanisms. And so we can categorize soil organisms based on their trophic guilds, their feeding strategies. And when we do that, and we can combine all of those methods that I've talked about up to now, we can combine all of those into what is probably the future of soil biodiversity analysis. Really, this is the kind of the cutting edge stuff that's happening, is food webs. And the food web concepts, of course, has been around for a long time, but now it's really sticking with soil ecology work. And by looking at the trophic guilds and the interactions between all these organisms and the strength of interactions, and tracing the flow of carbon and nutrients through the soil by using stable isotope tracing, we're actually able to start monitoring and building up interpretations of soil food webs under different management practices in ways that we were never able to before, and start to really understand the complexities and the nuances of soil food webs in different systems. And in fact, there's actually now an R package that has just come out by some colleagues of mine that you could look up that will enable you to do this in R. So just finally to wrap up, I'm going to run over by a few minutes, sorry, Kieran, but just to say there's a few monitoring initiatives that you should keep an eye out for in the UK that's employing a lot of these techniques. One of them is the Soil Biological Observation Network. So this is a global initiative that is looking at soil biodiversity around the world. There's still some countries which obviously we need to recruit, but the UK is a big player in this. So I was leading this in forest research, and now other colleagues are doing that, where we're taking soil invertebrates from and other soil measurements from around England and Scotland, extracting the invertebrates, taking soil physical chemical data, and it's all being contributed to this major project and being used for some of the image analysis that I mentioned earlier as well. So it's a big project. Keep your eyes peeled for that. There's also the Natural Capital and Ecosystem Assessment, which is a major UK one, or actually just England, but it's probably the most comprehensive soil biodiversity monitoring initiative that's taken place in the UK to date. And it's a collaboration between a lot of governmental arm's length bodies, Forest Research, Natural England, FERA, and others. And it's combining morphological identification with metabarcoding of lots of different groups and microbial stuff as well, along with soil physical and chemical data. And that's going on for the next few years. So probably, I think 2026, that might wrap up. And there'll be lots of interesting results coming out of that. And it's kind of a repeat of the countryside survey, or well, an expansion of the countryside survey, I should say. In terms of Europe, there's this cost action UDAFA base, which is worth looking out for. There's actually a call for data still, I think, for this. So they're trying to build a warehouse of Europe-wide soil biodiversity data. And if you contribute data to this, then you are I think you're a co-author of an atlas that they're producing. So it might be worth considering looking this up to see if they're still taking data. So if you already have existing soil biodiversity data, you should consider contributing to this. And finally, there's also, if you're interested particularly in Springtails, there's the Global Collembola project, which partners from all across the world have been contributing data to. And this is a Springtail specific initiative collating data from all across the world, different habitat types, and trying to build up a synthesis of Springtail habitat associations and other environmental associations. We've contributed data from the UK, and actually there's a data paper coming out early next year. It's just been accepted, and the database will be made publicly available for people to use as well. So there's a lot of, one thing I really like about the soil biodiversity community is they're very open-minded. We're all sharing data and making data publicly available, and everyone's working together pretty well, I would say. So if this is wet your whistle and you want to get into soil biodiversity more and take part in some of this monitoring, there's two things I would encourage you to do. Firstly, I'd encourage you to go look up the Global Soil Biodiversity Initiative, the GSBI. Their website is a treasure trove of really interesting information, and they try to be this sort of the axis between policymakers, researchers, and the public. So there's lots of resources there that you can go and look up. And they have been involved in some publications. So there's these two publications that I would encourage you to go and look up if you haven't already, and to start your journey in soil biodiversity monitoring. So the Global Soil Biodiversity Atlas, I think it was 2016, and then came the European Atlas of Soil Biodiversity. They're both really brilliant publications for finding a wealth of information about soil biodiversity. They're full of a lot of ecological information and really interesting maps of the world and around Europe of all these different organisms. You can download the PDFs for free online, so just go have a look. If you do order the hardback copies, though, I will warn you, there is no bookshelf on Earth that is big enough to accommodate these. They're bigger than A3. I don't know what the size is, but they're really huge. So be prepared to have a bit of floor space that you can put these down on safely. And with that, I will wrap up and take some questions. So I just say thank you very much for sticking with me to the end here. Sorry I overran a little bit, but yeah, thanks for your attention. If you want to learn more about soil biodiversity or you like some of my photos, you can find my website here, and I appreciate all the traffic. And just to say, here are the references that I referred to throughout this presentation. So thank you very much.